Thanks for coming. It's my pleasure to introduce Sergei, who I've learned from his CV. Started out studying cybernetics, and I looked it up, and I still don't know what that is. So you're gonna have to explain it. And then he went on to study applied math at Arizona State. And,
University of Arizona.
Sorry,
Big difference.
Arizona.
Big
Oh my
difference.
God. I always confuse the state schools. All right, sorry. Okay. Right. So what was I going to say? His group is really well known for developing software for inferring evolutionary parameters from sequence data, and I think it's fair to say has a specific interest in selection, and specifically in the context of pathogen-host interaction. And his group has actually done extensive work in HIV, and they were like really in a great position to kind of develop some insights into the same processes that we're all now interested in with SARS-CoV-2. And I'm really excited about his talk.
All right, thank you very much. Thanks for inviting me. It's actually exciting to travel again. I'm a little out of practice, but you'll see how this goes. I've posted a copy of my slides online, so you can just take a snapshot of this QR code or use the link, or I can share it later on. So I'll tell you today about the evolution of SARS-CoV-2, past, present, and future. Let me switch over to this presentation so it advances. So it's very topical. This Monday there was a New York Times article written by this great science reporter Carl Zimmer. He's been doing a lot of SARS-CoV-2 work that covers a preprint that we wrote on selection analysis of specific sublineage of Omicron. And just a quote from this, and I'll get to it at the end of the talk. So in a study posted online last week, an international team of scientists further deepened the mystery. They found that 13 of the unusual Omicron mutations were rarely, if ever, found in coronaviruses, suggesting they should have been harmful to Omicron. Instead, they were acting in concert. When acting in concert, these mutations appear to be key to some of Omicron's most essential functions. So it's actually a really good article. It explains things better than I can. Here's a SARS-CoV-2 genome. That's what we've been studying. We're actually, 7.5 million copies of this. You can small enough, you can fit it on a page. It's about 30,000 nucleotides. That's the reference genome. So the genomic features of SARS-CoV-2, for those that don't know, it's, in the grand scheme of things, it sort of sits in the middle of size. So they're DNA viruses that go up to about megabase. Some of the shorter RNA viruses are about 5 kilobases, and it's about 30 kilobases. It's long for an RNA virus. It's a positive sense, single-stranded RNA virus. A positive strand simply means it can be directly translated into proteins by host cell ribosomes. Negative sense is to do RNA transcription first. It's about 30 kilobases in length. It's one of the largest RNA genomes. It has some unusual features, including proofreading machinery, which is very unusual for RNA viruses. So effectively, its mutation rate is about 10 times slower than in other RNA viruses. It has other strange features like a slippage site in the open reading frame. It's programmed -1 ribosomal frame shifting, so it can control how much protein it makes. We have a paper on this, but I'm not going to talk about it. Allows the virus to encode multiple proteins in the same sequence and or regulate expression. The unique aspect of this pandemic is that's what I would call the real-time genomics pandemic. There's an unprecedented rate at which genomes are generated and shared. So the primary site for this is GISAID, which was an obscure German-run database for collecting surveillance data from influenza. They sort of have an unusual data sharing arrangement, which incentivizes data users to give credit to data producers, which is why I think people are comfortable depositing data there. But they're pretty restrictive about what you can do with the data. So you can't, for example, share alignments based at GISAID, which, so now we have 7.5 million genomes, probably 7.7 million today, because, it's about 50 to 100,000 new genomes are deposited daily. So what my group does and our collaborators study patterns of genetic variation, mutation, recombination viral genomes. And we use statistical methods and machine learning to try to understand what that all means. Here's an example of, sampling in the United States. So the color means of what fraction of infections have been sequenced. You can sort of see that in most of the country, it's less than 5%, especially in areas that have been heavily infected. And you've got places like Wyoming and I think that's what it is, yeah, and Colorado that are outliers because they have fewer infections and they spent more time sequencing. One of the things you realize when you collect 7.6 million genomes that contain errors and random mutations is that pretty much every single point mutation has been observed. So you get these silly claims when new mutations come up, oh, it's been seen in 5 sequences. With 5 sequences, you will see pretty much any mutation you can imagine. So that's not informative. You have to look at something that's a little bit harder than that. So here's, for example, a profile of sort of the number of mutations per genomic site, and what's cut off there is 100 sequences that contain the same mutation. So you can see that, a large fraction of the genomic positions have, variation in more than 100 sequences. So that is not very interesting. You can look at, among these mutations that people have been staring at and publishing papers about, you can sort of look at them in terms of their frequency. So how often do they, or you can call it AV or allele variant. Most of the variation exists at a very low frequency, under 1%. If you go up to high enough percent, so say, 5%, there's 100 of them. But they're obvious and they've already been extensively studied. So that's not something that's terribly interesting. And what I mean by extensively studied, so you can look at papers like this, which go to, just collect prominent mutations in viral genomes. So here's a cartoon of the spike gene with its various domains. So N-terminal domain, receptor binding domain, motifs, and so on. I find myself in a position I don't actually have to explain what any of this means because you've seen these are all the WHO variants of interest, variants of concern. This is their Pangu lineage designation. This is sort of a different, more detailed scheme for characterizing viral lineages. This is where they were first identified. And these are sort of the common mutations that have been observed in the majority of these variants. And you can see that a lot of variants share mutations in the same positions. There's this receptor binding domain, which is important for binding the ACE2 receptor and escaping antibodies. That's where a lot of variation happens. And then you can go on and build tables like this, where you take a particular variant and see how effective a particular monoclonal antibody, which is a column here. So all of these letters are specific monoclonals or combinations of monoclonals. One means it's sort of preference, it's neutralization against wild type, so the original strain. And if you see columns like this greater than 100, it means that, say, the beta variant, the gamma variant, the delta variant are completely left alone by this monoclonal antibody. It doesn't work against them. And you can sort of scan it around and see that at some point people have engineered individual mutations in the background to see that if SARS-CoV-2 genome gets this E484 decay mutation, it loses sensitivity to this antibody and so on. And you can do a lot more structural analysis to identify what they'll do. But my point is, the mutation is common, it's been studied, characterized, and we know what they do. But there's this sort of interesting box between very rare variants that could be errors, very common variants which we already know about that we don't know very much about, but there are too many of them to test exhaustively. So there's several thousand. So you're not going to productize this and do neutralizing screens against them because it's expensive and slow. This is where I think our tools come in, just comparative genomics, to try to sort of make sense of what they do. Let me just generally speaking of about viruses. Viruses are very interesting to study if you're interested in selection, because they're under a lot of different and constant selective pressures. Selective pressure is what drives variation and new variants, and you can see this happen quickly. So what are some of these selective pressures? So obviously, transmitting to a new host through a zoonosis when you jump species, or given from human to human, that creates sort of an environmental shock for the virus and gives it an incentive to change. Once it's inside the host, it's subject to strong multiple directions of selective pressure from the immune system. So you're going to have cellular immunity through cytotoxic T lymphocytes. You can have innate immunity and antibody-driven immunity. The longer this goes on, you start giving people drugs, then viruses are interested in developing drug resistance. They also compete with each other and try to become better in infecting hosts, so they have to balance virulence and transmissibility. If you're looking at longer time scales, so hundreds of thousands of years, you have this very slow ongoing evolutionary arms race between our protein, viral proteins that occurs. But despite all of this, like with everything else that's alive and functioning, most of the time, most of the viral genome is conserved. That's because it's doing well what it's evolved to do, and it doesn't need to change much. And most of the observed variation is neutral. and changes that are not neutral are important to detect early and ideally predict. So the analytical framework that we use is a comparative analysis framework. So we use substitution pattern analysis on large sets of SARS-CoV-2 data that can reveal genomic sites subject to selective pressure, which could be possibly important, both positive selection and negative selection. And we used a suite of stock and modified DNDS methods. So I'll give you a brief introduction to what they are, but I'm not going to dwell upon them. For coding sequence evolution that is developed by us and other groups over 2 decades and extensively used successfully in other RNA viruses. The methods are implemented in the Hi-Fi software package, which is what I've been developing since graduate school. We also have public servers that implement these methods at a website called datamonkey.org. One of the things we noticed is that as coronaviruses became a thing, we started getting more and more coronavirus submissions. So I don't have this graph, but like you can see the fraction of coronavirus data sets that go into our servers go from nothing to about 20%, right? And then it sort of decayed back to normal. So we spend considerable effort. I spent probably about 6 months of my time over the last 2 years on and off, doing software development and optimization and developing new techniques to deal with 7.5 million genomes. And all of this is a result of many collaborative efforts. So I'll present the results, but they were all done by a large number of people. So DNDS methods are really easy to understand conceptually. So you have mutation occurs at the level of nucleotides, which is shown here in the left panel. There's just some fragment of 3 different viruses: measles, rinderpest, and peste de petit ruminant. So these are 3 viruses that are related to each other. This is a nucleoprotein. And you can see there's a lot of nucleotide variation, right? You can sort of see 3 bands of this nucleotide variation, which corresponds to different viral species. These viral species are separated by thousands of years of evolution, so it's a long time period. Translate them to amino acids and you see no variation whatsoever. It's perfectly conserved. This is sort of a beautiful example that you almost don't believe would exist of negative selection. That means that any mutation that arose in this particular part of nucleoprotein, and a bunch of non-synonymous mutations arose because they're random, they never got fixed. So they were always removed because they're deleterious. So you have perfect conservation. Look at an antigenic site in influenza, hemagglutinin, which is a surface protein, one of the 2 surface proteins important for immune response in influenza. Similar level of nucleotide variation over a shorter time scale maps to a very high level of non-synonymous variation in proteins. So that's more or less what we do. This is obvious. If everything was like this, you wouldn't need any software, you just count, but it's a lot more subtle. When you start looking at big data sets, you got to do it in a systematic fashion. And this is an idea that goes back to the 1970s, where you use this intuition to sort of compare the rate at which synonymous mutations occur, normalized by some neutral expectation, and the number of fixed non-synonymous mutations divided by the proportion of random mutations that aren't synonymous. So they're sort of have a bunch of different names, but they're sort of known as DN and DS. Under neutrality, when there's no selection, these 2 should be approximately equal. So if there's no selection, then whatever mutation occurs has an equal probability of becoming fixed. So you can compute these ratios and compare them to 1, right? So if it's greater than 1 at some time, someplace in the genome, you're in for diversifying positive selection. If it's less than 1, negative selection, and everything else means you can't tell, right? So we call it neutral evolution. And a lot of it is due to statistical power. If you have enough data, you can always, find some subtle differences through neutrality. Nothing is perfectly neutral, I'll tell you that. People have looked for 100 years and there's nothing that's perfectly neutral. People declare synonymous substitutions to be neutral, they're not neutral. People declare introns to be neutral, they're not neutral. There's always, you can keep looking and always something has some subtle effect. So our selection analyses are based on phylogenetic probability substitution models. In addition to this basic DNDS, there's a lot of statistical trickery to make sure that it can accommodate biology. So selection varies from site to site, in a non-parametric manner, from time to time. We allow synonymous substitution rates to vary and so on. And we developed a data reduction technique, which I'm not going to really talk about. Practically speaking, you can only run these methods in maybe tens of thousands of sequences. In fact, there was an amusing PNAS paper that came out in the analysis of ACE2. It's a good paper. There wasn't one amusing statement in it. They used another software package for selection analysis called PAML, and they said because of computational challenges, we can only analyze 64 sequences. So, we can analyze 30,000 sequences, but it's still a lot less than, 70 7.5 million genomes. So to compress it down, and we developed some sort of somewhat principled techniques to select the most representative sequences and get them down to about 5 to 25,000 genomes, which we can handle. But that was a lot of work. Now you're getting from, at the beginning of the pandemic, we could maybe do 5,000 sequences. So going from 5,000 to say, 50,000, which you can kind of go up to 50,000, was a lot of work on my end, both algorithmically and software optimization. So let's talk about COVID. So what happened before COVID? Before anybody cared about coronaviruses. So the first thing to realize is there's nothing new under the sun. Coronavirus, SARS-CoV-2 is not the first coronavirus to infect humans and become endemic. It hasn't been fully admitted yet, but it's now an endemic virus. It's not going to go away. So it's actually a good thing. We wanted to go in the direction of the common cold virus. So prior to SARS-CoV-2, there were 6 different coronaviruses identified in humans. There were 2 endemic strains. SARS-CoV-2 now makes 5. There were 2 discovered in the 1960s, HCOV-C43 and HCOV-229E. They're different types of coronavirus. One is an alpha coronavirus, the other is a beta coronavirus. There's many millions of years of evolution between these 2. 2 were reported in the early 2000s, also one alpha, one beta. And then you had 2 epidemic strains, SARS-CoV-1, which is about 20 years old. It was a viral pneumonia outbreak, about 8,000 cases, killed about 700 people. And then you had MERS, which is also beta coronavirus, 2,000 cases, but it had very high case fatality rate, 35%. But it was very, very poor at human-to-human transmission, so it was contained. Now that would have been, a cataclysmic extinction level event if you have something that's a 35% case fatality rate. Based on the current estimates, SARS-CoV-2 is about 100-fold less fatal if you sort of aggregate across the entire population and multiple variants. So we actually had an obscure paper in 2013, but we tried to use. It was basically a methodology proof of concept paper where people were using these techniques called molecular clocks, which you will see, used in viruses, molecular clock dating, to try to estimate the origin of X, where X is measles, or X is influenza, or X is coronavirus. And because of a technical issue called saturation, where if you start looking at things that are too divergent, like you can see on the plot there, on the x-axis, we're sort of simulating more and more divergent sequences. We're talking about insanely divergent sequences, like 1 substitution per site, 10 substitutions per site, 100 substitutions per site. It's kind of difficult to visualize, but it does occur over these long periods of time. Traditional models are those little dots in the bottom that saturate and basically are not able to estimate anything that's over, say, 5 substitutions per site. We've come up with sort of selection-aware methods that can recover that degree of saturation. The reason for it is basically the only thing you can still align that's far separate is like that nuclear protein that I showed you, where all the variation is synonymous variation, and you have to really sort of dig deep into this to estimate divergence times. So, and if you look at, and we basically took alpha, beta, delta, and gamma coronaviruses from different species, little genomic fragments, and we estimated branch lengths into standard models, and you can see there's this subset of branches which you need to know how long they are in order to estimate how long it took the viruses to diverge, that they're basically infinitely long under the right model, and they're short and they're severely underestimated in the standard model. So the molecular clock dating analyses of coronaviruses prior to this said they're about 10,000 years old, which is just wrong. And it's a sharp contrast with the ancient evolutionary history of their putative natural hosts. In fact, when we do this correction, we found that they're probably as old as mammals, right? So they're tens of millions, possibly hundreds of millions of years old, which means that coronaviruses have been here since, we became a species, which means they have adaptive features. Other interesting thing is that nobody cared about this paper until coronavirus came around, and then all of a sudden, like, oh, coronavirus, let's download and look at it. So let's now move on to actual coronavirus that we care about, SARS-CoV-2, and there's this time scale.
Sergei,
Yes.
On the model, you always will assume that there will be a slope, and that slope should be more or less a constant.
No.
Or would there be a condition by which you actually will observe saturation because basically the mutation sort of saturates space?
So the question is, are these models able to sort of go as far back in the past as you need to go? No, they don't. They just go further back than the standard models. And they will tell you that things are saturated by giving you an infinite branch length instead of giving you a branch length that's 3, but it really should be infinite. So basically, sort of the main advantage is it's going to tell you if something is so ancient that you shouldn't be able to estimate how they are. It'll give you a lower bound It's not going to estimate it accurately, but it's basically going to tell you that it's infinitely old, which is better than saying. So what precipitated this, and just as a side, there was a paper that published that measles is 600 years old if you do molecular clock dating. There's a problem with it because there's a text in Arabic that's 1500 years old that talks about the differential diagnosis of measles and smallpox. So you know that your molecular clock estimates are at least threefold. They're directly contradicted by historical record. So that's what drove that model development, which then applied to the coronavirus. And then people said that influenza was 3,000 years old. No, influenza is not 3,000 years old. Influenza is also saturated. It's infinitely old. We don't know how old it is, but it's older than 3,000 years. So here's the actual timeline. So if you remember, there's actually a significant debate, which I'm not going to talk about when SARS-CoV-2 entered the human population, sometimes in 2019, but there's a big uncertainty window on when exactly. But let's start with 2020. So zoonosis early adaptation, we actually had a study that looked at what happened to SARS-CoV-2 during its emergence from an animal progenitor. So was there any selection of the virus before it got into humans? And was it sort of where it was? So in order to do this, we collected all closely sarbecovirus. So these are beta coronaviruses of a specific subgenus, coronavirus, sarbecovirus, and defined a specific clade, which we call the novel coronavirus clades, of all the bat isolates and pangolin isolates at the time that was most closely related to SARS-CoV-2. So it kind of looks like this. So we focus on selection recombination history of this clade, and specifically the branch that leads to SARS-CoV-2. So there's a tree, there's a bigger tree that I'm not showing that includes other more distantly related sequences, but you might recognize some of these. So there's like RAD-G13, which was the first bat strain that was closely related, this Yunnan 02 strain. There's more of them now. We have an updated analysis, but nothing has really changed. So we found selection of extensive recombination history in the novel coronavirus clade, which you will see probably as the endemic progresses, you'll start having recombinant variants. We'll also have an example of this happening, but I'm not going to talk about it. So later studies actually show that this recombination is not randomly distributed across the genome, but there's specific hot and cold spots, especially around, seems like, relevant parts of the spike gene where recombination events tend to happen more often. So here's sort of a summary of what we found. So here's a viral genome here at the bottom. And the boxes are showing you non-recombinant fragments. So every time there's a break, like here, there's a recombination breakpoint here, here, here. And you can see there's a whole bunch of them in spike. There's 5 fragments that we found in spike. So this is all from published work. So this was a PLOS Biology paper in 2021 that's referenced somewhere. There's also a compelling argument that you had sort of a genome-wide adaptation in nucleotide composition. So there was a CG depletion. That primarily has to do with host environment. If you jump into a new host, especially if you transition, coronaviruses can be either respiratory or enteric. When they switch between those two tropisms, they usually significantly change their GC composition because they replicate in a different environment and they sort of to go in different hosts. Overall, this black line shows our perfectly conserved amino acid sites, right? So the vast majority of genomes in bats are conserved. There is some primary evolutionary signal is negative selection, and there are a few sites under positive selection in the novel coronavirus clade, and some segments are under selection in the novel coronavirus clade. But there's no evidence of selection on the coronavirus, on the SARS-CoV-2 branch as it is. So evidence of selective pressure in the nCoV clade, but not in the SARS-CoV-2 lineage. So here are 9 segments where we found some evidence of selection. The selection is depicted by a colored branch. So this is SARS-CoV-2, and if you just scan across, you can see that selection operated various parts of the genomes in the past in this clade, but not in SARS-CoV-2 itself. So it appears to belong to a generalist virus clade, adept at host switching and not requiring significant immediate adaptation during zoonosis. So it looks like once it got into the human population, it didn't experience immediate adaptation immediately prior to that and didn't do much after that. So imagine what happens. You have a new coronavirus. It spills into a population of naive hosts. Nobody's seen anything like this. There's no immune response. It's adapted to spread. You would expect it to just spread. Nothing was going to happen, right? Until it starts hitting some immunity or some selective pressure. And for the first 9 months or a year, up to October of 2020, that's exactly what we saw. We saw mostly a neutral pattern, We're naive hosts, no selective pressure, so the virus just drifts across. You have some mutations fixed. And if you go back and look at literature, people were basically scrutinizing every single mutation because it wasn't very much. Maybe this one matters, maybe this one matters, maybe this one matters, but really nothing mattered for the first year. There was one, this D614G, which could have actually been an ancestral variant for all we know. Just, there's the situation is muddled. And there's relative lack of meaningful diversity. So here's, just another way to view this variation where you have, sequence more and more genomes, you will effectively have almost a perfect, almost every position in the genome is going to have some variation among hundreds of thousands of sequences. But if you look, for example, at what fraction of them show any evidence of selection, and this is a square root scale, it's almost none. So it grows much, much slower. You accumulate variation, but most of this variation is meaningless. So only a few sites exhibit any signal for positive selection, and I'll come back to this in a second. So this was all fine, but unfortunately, coronavirus tends to do things around the holidays. So this is the second year, it sort of ruined my Thanksgiving and Christmas holidays. This was a Christmas holiday present. In 2020, you had 3 independent emergences of what was called N50Y lineage. So N50Y is a characteristic mutation in spike at position 501, which was later implicated in enhancing ACE2 binding and maybe changing molecular dynamics. There were 3 of these lineages that arose. They weren't called alpha, beta, and gamma at the time. They had numerical designations, B117, 1352, 351, and P1. But now in the WHO designation, they're alpha, beta, and gamma. And there's a striking pattern of shared changes. If you try to look at, for example, here, there's a stack of 3 mutations that are shared by all of them. So 1 mutation that's shared by all of them, and a fair number of mutations that are shared by both. And there's this deletion that's shared by 3 of them. So there seems to be like there's concordant changes going on. And they were all independently derived. So one arose in the UK, one arose in South Africa, and one arose in South America. You put them on the phylogenetic tree, they're unrelated. They're all separate. So it's not like these variants developed and then were further diversified. No, they were all developed separately. So our question was, can we find any evidence of convergent evolution by doing large scale data mining? And this was published last year in Cell. So we have a paper, it was basically, Cell makes you do a graphic summary, which is great. So, and this is all credit to the primary author and my great collaborator from South Africa, Darren Martin, who drew this. So, we have this is an indication of what happens at specific genomic position in the viral genome. So you have, some genomes with E, some genomes with K, and there's just some variation, but not enough for us to detect this as a positively selected site. Now, now you split these things into different lineages as they emerge. And all of a sudden you see that, this lineage fixed the substitution from E to K, so that's all you see. And this lineage fixed the substitution from E to K, so that's all you see. Now you can detect this as a selective event, right? Because you had multiple substitutions in the tree, and there's enough there for our statistical methods to pick it up. So an excess of independent mutations that change the encoded amino acid triggers a positive selection signal. And this was actually, when we retrospectively looked, as I'll show you, this was evident before the variants emerged. So discovery of the alpha, beta, and gamma lineages is apparent that K is favored over E at this site.
Sorry, how can you rule out contamination? I mean, they are different part of the planet, but they could travel and somebody, so could there be
well, if you put them all in the same tree and they're related, right? If you believe phylogenetics, they should have some degree of, common ancestry, but they sit in 3 separate events. And there's actually more evidence that not only do they sit at 3 separate clades of the tree, but there's sort of, for 2 of them, you have this pretty convincing pathway of stepwise substitutions that lead to it. Alpha is strange. Alpha just emerged, right? It had like 8 mutations that had never been seen before in the UK, which was very densely sampled. And there was sort of, at the time it was hypothesized that this variant arose in one individual that was chronically infected for 6 months or so with SARS-CoV-2, and that all evolved within one person and then transmitted on. So they even had different routes of development. It's pretty, we're pretty certain they're unrelated. So, and you can sort of, there's a sort of busy plot, but I just want you to focus on this thick line, which kind of indicates one measure of selective force driving the evolution in spike over time. And here's, there's a ramp up of selective pressure that occurs in the fall. And it occurs in a couple of genes, but most importantly in spike. And here's a different picture that sort of shows you that you can detect things before they become a problem. So here's a plot. There's a time series here. The x-axis says, it's an analysis. So what's shown here is a series of comparative analysis of SARS-CoV-2 genomes for evidence of selective pressure in individual sites. A time point means that if it's November 2020, it means that we analyze sequences from November 2020 and 3 months prior. So the sliding temporal window analysis, we'll march across the x-axis. We're sort of following 3 months intervals in the viral database. Here are all the sites that were mutated in one of those 3 lineages. So there's 501 spike and 484. And if there's a red circle, it means that it's at that time point, our comparative analysis were showing that the site is under positive selection. This gray line is when the variants were detected. And you can see that for a fair number of these variants, including these mutations, including 501, 570, and so on, 681, which you care about, we could detect evidence of selection several months prior to their emergence. So there's an evolutionary signal that precedes the emergence of variants. So multiple clade-defining mutations were detectably evolving before they became clade-defining. And then once you have a selective shift, now you have a split. And you have 3 parallel evolving lineages. So here at the site K is fixed. But now what you start seeing is in this alpha lineage, I'll get you in a second, some of the sequences are also exploring this K. So as you get evolved, it looks like this lineage is also trying to see what happens if it goes to K. So this is a canonical pattern of convergent evolution, right? Where the same things are occurring in multiple different lineages. Yeah, question?
If you notice a particular thing is going to be positive selection, can you be for sure positive that a new lineage will occur?
No. I'll close it all out with what we can predict at the very end. So we used data up to February to define 35 convergent sites between 3 lineages. So we basically productized this analysis and tried to find patterns where, there are convergent changes in multiple lineages, sort of like this. Don't worry about it too much. And then we used data up to June to validate convergent sites. And you basically get things like this, where these sites were detected by our analysis as convergent, and you see a couple of things. You see, so this is an evolution at a particular site. Again, time axis here. This is the time of emergence, so November of 2020. A red circle means this site is now under positive selection. And this gray line means what's the global prevalence. So if it grows up higher, it means this mutation is becoming more and more prevalent. And if there's positive selection, for example, here, this mutation is not prevalent. It sort of stays at about 10% frequency, but it's sort of constantly generated at a low level. So we found 20 out of 47 in the analyzed convergent mutations that contributed to the fitness of 501 lineages, more than doubled in frequency at least one of the lineages between March and June. So, remember, we used training data to February, then we used this to predict which sites will change, and it did pretty well. Right, and you can sort of look at, actually, I'll skip that. It's not that important. So this was all fine and well, and then, alpha, everybody, last spring, everybody was saying alpha was going to be around. This is the dominant lineage, we'll have to worry about. And then, by late spring, summer, alpha disappeared. It was displaced by delta, which did not have the 501Y mutation. It had this S5452 mutation, which appears to be in a different fitness peak compared to the S501. And it just as a little aside, I'm not going to spend too much time on it, but there is actually evidence that some of the same mutations that we found in the 501Y lineage were being kind of tried, generally discarded by the delta clade. But these are still sort of mutations where you would expect, where we saw things occurring a lot more frequently than we expect by chance. And in fact, some mutations, so for example, 138, which was one of our meta signature mutations, was observed in about 40,000 delta sequences, including some of the weird ones that either contaminated or recombinant, so up to 6 mixtures, doesn't matter. All right, so Omicron emerges. So we don't, it's difficult to be the dominant SARS-CoV-2 lineage because it's a short and fraught rain, something's going to displace you. It's just a question of when. And Omicron was, again, it emerged the week of Thanksgiving last year. So again, we did a lot of work in December on this. It was a very strange lineage. So here's a little evolutionary tree of, all these red branches are all the other lineages of concern. So alpha's here, delta's here, beta and gamma's here. And here's Omicron. You see kind of two things. One, well, it's actually two. This long branch of BA2 was a sublineage of Omicron that wasn't originally discovered. So if you delete this branch, the original branch was actually twice as long. And it comes out of nowhere. There's nothing else in these 6 or 5 million genomes at the time that looks like Omicron. Where the hell did it come from? So no relationship to Delta, and it pretty quickly displaced Delta. Lots of missing evolution. The missing means we don't have any intermediaries. And this is the most densely sampled pandemic in history. Where did it go? How did it do all of this without being seen? So lots of strange mutations in spike as well. So I'll develop this. So we set out to figure out what we can tell about it only using comparative evolutionary data. And the reason for this is it takes about 2 to 3 weeks to do any lab characterization of what it does in terms of vaccine, just neutralization titers. Take a couple of weeks to run. There were other problems because there were actually primer mismatches in some of the key parts of Omicron. Some of the lab assays didn't work. So we had about 3 weeks to figure out, to try to make a prediction before somebody did validation. So I wanted to do this. What can we do just based on evolutionary data? So here's a picture of where all the Omicron mutations are across spike. I'll come back to this in a second. Just take away from it, there are a lot of them, a lot more than in any other of the variants. What is unusual about some of them, so some of them we've seen before, so 501Y, seen before. All the red ones are the ones that at some point had been previously selected in viral sequences. So we would expect variation to occur there because the virus has tried it before. But these blue sites are very strange because they were negatively selected in the past. So there was variation. It was kind of like that nuclear protein that I showed you in measles, where variation was coming up, but amino acids were preserved. So negative selection implies that individually, amino acids at those sites are likely maladaptive, right? They arise, they're removed on their own. So why do we have all of them together? And we've also applied selective pressures to pre-Omicron sequences. So, these are all, each of the rows in this plot is a signature site in Omicron across the entire genome. And the same picture as before. So, and this is using pre-Omicron data. So you can see that a fair number of them showed evidence of positive selection, extensive, some of them going all the way back to the beginning of the pandemic. So red means positive selection, blue means negative selection, blank is neutral. But like I can zoom in at these sites, so 339, 371, 373, and 375, they were all negatively selected, right? No evidence that the virus was ever doing anything to change amino acids there. So what's going on? We've also then decided to see if that existed, a variation like that existed in the more distant past. So look at the evolution of these positions in close related sarbecoviruses in bats. So we're talking about a century of evolution, give or take. So most of these mutations are never or rarely seen, with a few eliminating exceptions. So this is a cartoon tree of sarbecovirus sequences at position 339 in spike, which is where you have this negative selection. There is a change that we observed in Omicron on this branch. So sarbecoviruses are here. So SARS-CoV-2 is actually in this clade. So in this distantly related clade, and this clade, these bat viruses do not use ACE2. So that's a very different phenotype. They use a different cell receptor for entry. And it's not dispositive. I mean, obviously there's a bunch of other mutations there, but it's kind of interesting. And then if you look at some other mutations like this pair 493 and 505, the same pair of mutations was observed again in a somewhat distantly related clade, and they occurred at the same time. And this is a key, when you have these coordinated mutations, this is a canonical signature of epistasis, right? Which means that things are evolving together interactively. Right? That's pretty much, if you wanted to look for epistasis using comparative sequence analysis, this you would do. You would map substitutions into a tree, and it would use a statistical tool. We have one based on Bayesian graphical models that just computes the likelihood or the probability that they're not conditionally independent, right? And you can do this. So it points to the potential functional impact and epistatic interactions. A few of these mutations have also occurred in other human coronaviruses, and one even showed evidence of convergent evolution. So, but mostly not seen before. So, for example, here's a cartoon. The last column shows if there's something there, it means that this particular mutation was seen as in that human other coronavirus. So, HQ1 and so on. But again, it's sporadic. And at this point, there's so much annotation that I can throw you a challenge and say, here, 15 random mutations in spike. Go find what they mean. And you can look through annotation and you'll find pretty much any one of them found somewhere in some in vitro experiment, some frequency in genomes. So the fact that an individual mutation was observed before is not particularly informative just because there's so much variation that's already there. So let's look at sort of multiple order arrangements. So the effect of multiple mutations together under epistasis is very different from the added effects of individual mutations. That's the definition of epistasis, right? You can have two mutations that are individually maladaptive, bring them together, they're now adaptive. If you've never seen this before, haven't thought about it, the classic example that I give here is compensatory mutations in HIV, where you give HIV to a person, put them on a monotherapy of a drug, HIV is going to develop a drug resistance mutation. That drug resistance mutation happens in an active site of, say, reverse transcriptase or protease, so it comes with a fitness cost. The virus is now less able to replicate. So you let it run around for another 2 weeks in this individual, or 3, you're going to have a compensatory mutation that comes up to restore the action of reverse transcriptase or protease. On their own, the second mutation makes no sense. It actually wouldn't occur because it doesn't need to happen in the wild type protease or RT. With a compensatory mutation in the presence of a primary mutation, that's what I'm, and you see this pattern in the evolution, chronic evolution of HIV in, viral sequences, say HIV in a chronically infected individual. The antibody selective pressure of the immune system keeps pushing it, and he sort of, it pushes it, the virus changes, then the antibody catches up, it pushes it some more, and it changes. So you have this nice consecutive development of mutations. But perhaps these, Omicron mutations did co-occur previously. And you can sort of, we looked again, we have this massive database, so just look at pairs. And if you see a pair that's red with a big circle, it means that they have occurred before in the past, right? So it's literally just a very dumb test where you see if pairs occur more frequently than you would expect by chance, given conditional probabilities of each mutation occur separately. It's the same thing as, it goes behind like Blows and scoring matrices. So by and large, you see a few red circles, but there's not a pattern of co-occurrence of these mutations. In fact, a lot of them would be actually opposite, right? These mutations do not occur together. They occur less frequently than you would expect by chance. So we've never seen these combinations before. So they did not. So 13 mutations occurring in 3 spatial clusters that are individually would likely be maladaptive. So how can they arise? So first, I can show you where they are. So here's a cartoon of a structure of the spike trimer bound to human ACE2 receptor. And these are the sites in these 13 mutations can be partitioned into different clusters. And they sort of, I'll come back to this, but just remember this, they're in 3 separate clusters. So how can they arise? And our hypothesis, that I haven't seen a better one so far, is that consistent and different selective pressure, probably in a chronically infected host, possibly in a different animal host, have done 2 things. One, they allowed Omicron or the progenitor of Omicron to spread undetected. We don't test in animals much, and we don't look systematically for chronically infected people. We sample acute infections pretty well, including in South Africa, but we don't look for these reservoirs. And the longer this goes on, a reservoir is somewhere the virus can hide, evolve diversity, and come back to humans without us knowing. So I think that's what happened here. We don't know definitively. There are anecdotes that there are about 20 cases of chronically infected hosts that show similar evolutionary patterns. Not quite as extensive, so we don't have a full story yet. And there's also 2 lineages, but it had to have occurred at some point in a hidden, evolutionarily different environment. And the 2 most logical ones would be chronically infected host or something like rodents. The reason rodents are popular is because the Omicron spike carries mutations that allow it to bind to rodent ACE2. And of course, then you have people saying, it arose because of mouse adaptation through lab passaging. You put this in humanized mice. And my answer to this, there's a paper from 2020 that showed that all you need, there's actually a mouse adapted strain, SARS-CoV-2, that you can buy. It's a bio, it's a reagent. You need 3 mutations in spike in order for this to happen. That's all you need. You don't need 30.
The ferrets is more tragic, right? The story of ferrets is more tragic.
Ferrets die. Question?
It's a weird question. Has this ever happened like historically where you get something that just didn't seem to have any like relative or tree Omicron out of nowhere? Historically, this has happened before or?
Oh, it happens before all the time, but we have very poor sampling of everything else. So you could just say, then you can explain it by we just didn't sample the relatives. Here we spent billions of dollars, 2 orders of magnitude, maybe 3 orders of magnitude more sampling, and it's still missing.
And is there a decent hypothesis for why this might happen?
Well, so it just basically evolves somewhere we're not sampling, right? So it's either in a chronically infected individual or in a different animal species we're not looking at. All we're doing is basically just doing normal epidemiological surveillance.
So for sure, it's because we didn't sample properly.
For sure, there's just no way it could have evolved, could have escaped sampling.
Okay.
I mean, if you do this, if you do molecular clock estimates, it goes back to summer of 2020, which means it was circulating for 18 months. It's super infectious. Once it got in, it displays Delta within a month. So you start thinking about just put all of these things together. It had to spread for a significant amount of time. It was transmissible for some time, right? It wasn't a question of one mutation. So you would have seen it a lot faster. You would have seen it somewhere. You would have seen it in South Africa and so on.
So you would have seen it until the functional mutation happened, and only then it triggered. It could have been actually neutral, or it could have been.
But then you would,
with Delta.
at some point you got to say, that only works if all of these mutations were necessary. But the interesting thing is that what you see, what I can't really explain is, and I think we need more data for this, is that you could potentially explain it with one variant, but you actually have multiple variants. So you almost have like this half evolution goes to Omicron BA2 lineage, which is now increasing in prevalence in Europe. And then there's another big step to BA1. So just, so you already had like an intermediate variant that was apparently transmissible, better than Delta, and we still don't see it. So
you better than nobody and anybody else would know that if you have a switch, say a given mutation that all of a sudden disrupt or neutralizes a given certain set of antibodies, right? That can actually trigger a whole bunch of, functional mutations on the fly because everything is mutated. Right?
yeah, but it's just the question, and you're right. I don't actually, I haven't put probabilities on it, but the fact it just back of the envelope calculation in terms of how quickly it grew once it emerged. And how much sampling, and South Africa is very good at sampling. So it's not like a blank spot. It's not like, some of the Central African countries which don't have a lot of sequences. So just very difficult to imagine it was there for a year, circulating in humans with no detection of anything. But it's also interesting, now that you mention it, one of the amusing side stories is that so once Omicron emerged, people went back and started looking at sequences that were Omicron sequences in GISAID that were deposited in October. So people had Omicron in October. They just didn't realize it was Omicron. They simply just do batch deposition of sequences, but that's as far as it doesn't go past that. So we missed it for about a month. And the reason it was found is because it started growing up in frequency in one geographic location. Which I'm going to pose you a question here now. If with this level of surveillance, this level of detection, this level of attention, we had Omicron sequences in October. We didn't know that we had Omicron sequences. Nobody looked. How likely do you think that the original SARS-CoV-2 variant was detected immediately after it got into the human population in China? When nobody knew what to look for, it was sort of a generic, infectious disease. So it just, food for thought.
One thing I still don't understand, I'm confused though, is there's a lot of mutations for Omicron, but none of them have co-occurred before. So it's like they always luckily happen together at the same time.
No, it's not at the same time. It's like, so what's, if you're doing evolutionary experiments, what's the easiest way to generate a bunch of mutations? You do serial passaging. You put something in the same selective environment and you sort of just turn the wheel. That normally doesn't happen with coronaviruses because they don't, once you jump from person to person, two things happen. You have you have population bottleneck on transmission. So you have some randomness that removes variation. And when you go from one individual to another, you're going to have a different selective environment. So the virus, instead of going on the gradient in the same direction, pushed by the same force, goes sort of in opposite directions. And you also just randomly remove things from the population. And where this will happen is when you go into a chronic individual, that's exactly what happens. You have constant turnover under constant selective pressure. That's how chronic infections establish, because the immune system of the host is not able to clear it. So it's but it's still responding to it. It's just driving it to keep making changes. And the same thing will happen if it goes into a new host. And all of a sudden, it's not fully adapted to, say, replication or transmission between, rodents or whatnot. Question?
Yeah, so let me see if I can get this straight. So are you saying that if you are doing basic surveillance, right, and you see even just one individual, right, with many mutations in the SARS-CoV genome relative to the baseline, that alarm bells should start ringing, like, oh, this has been around in a patient and it's likely to be transmissible, and then in 3 to 4 months, it's going to be dominant and we should start thinking about how to combat it? Or are there also cases where that many mutations occur, but it doesn't end up, let's say, taking hold?
Yeah, so most of the time this happens as a contamination, right? But you can remove contamination, because if it's contamination, you'll expect a mixture of two known strains. Mm-hmm. If you have something that's a combination of rarely seen mutation, especially if it's replicated from different labs, then you start looking. Because now people started looking and said, oh, there's Deltacron, right? The combination between Delta and Omicron, it's all fake. It's all lab contamination.
Okay.
So, can I follow up on
Sure.
the question? So I think maybe this is what you're getting into. So presumably this situation where you have chronic infections in some immunocompromised individual, they happen fairly routinely, and you end up with something that looks really divergent. Is the typical situation that now it takes over, or mostly that would die out in the general population?
The latter. I mean, you can just, right. So the billions of people have been infected with coronavirus, right? For sure. There are tens of millions of people that have HIV. There are probably millions of people that have uncontrolled HIV. Mm-hmm. So there must have been at least, tens of thousands of cases like this. And just because we don't see these variants emerging all the time, that gives you that this is a rare event. And you would actually expect it to be a rare event. So the one thing that doesn't quite follow from this is that once you follow a selective gradient in a person, you're very well adapted to that person. But that comes at a cost, because usually now you're not adapted to transmission. All you've done is you become very good at avoiding that antibody response.
It's the opposite. If you see one of these things in a database, it's almost like it's even less concerning than
but if you
diversion, because you're like, this is adapted to some sort of really weird
but if you run this experiment enough times, go to a casino, at some point you're going to draw a royal flush if you do it enough times.
I guess, so like you said, so if you're in this person, it's well adapted to that person. But if you'll go out a bunch of people and you see the same simpler pattern of those things occurring, right? You have like some mutation,
yep.
But for each person, you're adopting more, right? But if you can only stick people and you look at a simple pattern that's there with extra stuff for each person, and if possible, that's a strong.
Yeah, I mean, if you have sort of steps, evolutionary steps. All right, let me just. So one of the interesting things is that this preprint just came out this week that gives a structural basis to what one of these clusters does. Apparently, it stabilizes, a receptor binding domain interactions and actually makes it more stable in a particular configuration. Well, that's kind of cool. I mean, we could just make general hypotheses, because when you see things like this, it has to imply some degree of epistasis and functional adaptation. And if you actually look at all of this, again, this is a picture from the New York Times. So, this has now been shown to stabilize, receptor binding dynamics in a particular configuration. So this cluster 2 enhances ACE2 binding and has escape mutations against several classes of monoclonal antibodies, except this one too. And this seems to be involved in the fact that the Omicron variant. So coronavirus can enter the cell through one of two mechanisms. It can do direct cell fusion, which is the canonical way for SARS-CoV-2, or it can do endosomal entry, where it gets carried into the cell through an endosomal route. So Omicron is actually much more, typically viruses just do direct fusion, most of them. Omicron can use both. So that talks about, the fact, cleavage dynamics differences and things like this. So finally, and this is going to be very quick, but this is very different. So all of this is retrospective. Something happened, and we try to figure out why it happened and what it needs. Can we actually do anything looking forward? And there's a paper that just came out again in Side Translational Medicine with, this was mostly driven by some data scientists and my old HIV colleagues at Vir, which is a big biotech company. Their antibody is the only one that still works against Omicron. So they, part of the reason I argue is they're actually thinking about comparative evolution. So instead of simply figuring out what the virus has done, can we make any predictions for what it will do? So can we predict? And I think the difficult question, we cannot answer difficult questions. We can't tell you which variant will outcompete the other. Nobody would have told you that Delta was going to outcompete Alpha. Nobody would have predicted Omicron. We can talk generically that something will occur.
Of course, I can.
Well, yeah, but it happened, right? And with so many predictions, somebody's going to write. So let's simplify the question. So short-term forecasts of which of the circulating mutations will increase in frequency. So one mutation, given what it does today, use whatever information you want. Is it going to increase in frequency in some sense in the near future? So with this paper, we evaluated a large number of sequence and mutation features in terms of their predictive value. And this is your standard, machine learning setup where you have training data set, which is sequences up to this point from some time window, say the previous 3 months. You take the features from this, you train your model and try to predict what's going to happen in the next 3 months. And you do this every month, right, to update your forecasts. And you also have to define your outcome. And our outcome is a spreading mutation means it significantly increases in frequency. So you have a significant p-value in the 2x2 table. And then it has a fold change from baseline of at least 10 in at least one country, and a fold change of at least 2 across 3 or more countries, and a minimum global frequency of 1 in 1,000. Right, so that's what it means for a mutation to spread. This is just a long list of features that we've tried, evolutionary features, immune escape, immunology, transmissibility, and even some weird stuff for natural language processing. And it works remarkably well. So here's an example of, detecting. So if you just follow one of these curves, take any of these curves, say, maybe this one, so the dotted means that at this point, our model would not have predicted that this mutation is spreading. And then solid means from this point on, it was predicted to be spreading. So you can kind of see, for instance, this pink line, it first is, then this is K417N, which went on to be a prominent mutation in the beta and delta lineage. It would have been first detected by this method in September of 2020, when it was circulating at a very, very low frequency. And, and if you actually, sorry, did I miss? I'll come back to it. Sorry, I got my slides out of order. I'll actually even skip this. So most mutations that eventually spread could have been predicted several months in advance. So this is sort of the number of mutations that eventually became spreading. For most of them, they could be predicted about half a year in advance.
Is the method similar to the way we predict the influenza?
No. The beautiful thing about it is it has a 3-dimensional predictor, and the predictor works by ranking the score. Right, so it's about you can compute it in Excel. It doesn't require. It's about as simple as it gets. It's beautiful because with this much data, you should be able to use very simple predictors. But here's an example of what you might do, right? And this is, I think, part of the reason we should be doing things like this is that so you have, say, a collection of therapeutics, monoclonal antibodies, right? You don't want to wait for a mutation to arise that's going to kill these monoclonals before you do something. You want to try to predict which mutations will rise in frequency, test beforehand, and make your decisions about whether or not you need to look for new monoclonals. So here's an example. For example, one of the predicted mutations, position 496, I think, the Vir folks actually predicted it and went on and did some testing and showed that a bunch of antibodies, monoclonal antibodies would fail against it, but not theirs. So presumably, if you're a drug developer, you want to have a pipeline like this to sort of churn it through and say, is my drug still going to work? Yes or no? And if it's not going to work, maybe change it. All right, sorry, I'm running a little bit over.
Simple predictor, you didn't tell us what it is.
Oh, the predictor, sure, I can tell you what the predictor is. The predictor is, I can explain it. Basically, the most fundamental predictor is that you take a specific mutation, right? And you see how many unique genomes does it occur in, right? So you can have a genome simply means a complement of mutations that defines a viral strain. Many of them will be represented by multiple sequences. So the most common, say, haplotype might be represented by 10,000 sequences, but it counts towards one haplotype. So the most predictive feature is what fraction of haplotypes, unique haplotypes, does your mutation occur in?
What's the direction?
The more the better. It basically, you can view this intuitively as it's a proxy for multiple emergences. It occurs in multiple genomic backgrounds. It's a proxy for sort of tolerability. So, if you need a specific genomic configuration for it to appear in, it's going to be rare. That's all it is. I was happy because my evolutionary measures did pretty well. I mean, it was the second best, just, sites under selection, but it's a lot more expensive to compute.
So it's this actually the single score predictor is the predictor.
You can throw in geographic spread and you can throw global frequency and normalize it. That's all it is, a three-dimensional predictor. And the way you rank it, you compute that score, you rank the mutations and you sort of cut at some rank. And then it gives you an AUC of 0.9 or 0.95 or something like that, depending on where you look. So big data, dumb inference, great. Central limit theorem applies. I like it. I would like to acknowledge all of my colleagues from multiple places that did a lot of work for all of this. Just like I mentioned, I'm on a lot of papers, but I did little pieces here and there. I'm just sort of doing a synthesis of what everybody else has done. And this has truly been a sort of an international collaborative effort. And, we'll continue looking. Thank you. Andres.
Yeah, so which mutation is next?
I don't know. I was too busy looking at Omicron. The Vir people recomputed their scores. I don't know what they are. I haven't looked.
Okay, all right, I'll be worried about that.
Carlos?
Yeah, so, well, I mean, the FDA will kill all the predictions, right? Because the time for approval, a year will be too late for this early warning. But something that seems to be missing here that I was hoping to see is the classification of the origin of the specific mutation from evading immune response, from functional, from evading natural immune response, evading antibodies, right, and so on. Is there any effort in your part, or
No. Other people do this. I mean, you can productize this and do lab assays. You can measure neutralization escape directly. What we do is we sort of try to, the whole focus of this, I think, sort of our ultimate goal is to do something that's proactive, to do something that's somewhat predictive. And all of this is building like positive selection signal is important because you can see this before things occur. Things, epistasis kind of is going to show you that some functional reconfiguration occurred. But considering how many people are working on the wet lab side of things, they will do this. They will do the binding, they will do the neutralization assays. So I'm not doing any of this because one, I don't know how to do this. And then,
the marker should be also in the sequencing, right? Because the rate of the change, the rate of a mutational rate will highly depend on where they come from and or what purpose they fulfill. I mean, I wonder, no?
Well, I mean, there's just general structural conservation, but I mean, it's really difficult to predict because each of these mutations, as I hopefully showed you, will have different effects in different genomic backgrounds and different environments.
Right.
So you're not capturing all of the relevant covariates, right? Just sequence variation can mean different things in different contexts.
Okay.
But yes, I mean, there are I think there are two sites in Spike that don't have any amino acid variants in any of the sequences. I don't understand why that is, because just randomly you should have seen an amino acid variant. So it's probably some sort of a sequencing artifact. But you can start looking at things like this to try, to find.
Yeah, interesting.
Sorry, there was a question.
Yes. So I'm a PhD student in computer engineering. So my question is about the project management. I mean, this risk area that is very timely and a lot of interest. Right? You mentioned working on sensitivity, these papers are published very recently and very intense. So how do you do really manage to get sleep is to look still healthy? And the Nature have accelerated article for this area. It seems it's a great opportunity we can jump in, but I think I will worry whether we have enough energy to really do some research.
Well, I mean, all of some of the bigger studies, there were 10 or some cases 50 people working on it. So you sort of do little pieces. And the practical way is just you focus. Unfortunately, there's not a whole lot of time to do new development. I try to carve it out. So you just basically just, crank the wheel and things that you already know. And I took some time last year to, during the lull to do some software development and make sure we can scale. But yeah, there's always too much going on. But yeah, it's always like, my South African colleagues work at night, when I sleep, they work. That helps.
You have these examples where, you're usually tracking like positive versus negative selection over time. Do you have any examples where you can show differences in positive versus negative selection in space at the same time? Like
Geographically.
Yeah, geographically. Like, so for example, like Australia had this huge epidemic in an infection naive population.
Yep.
At the same time, when that was the case almost nowhere else in the globe. Can you see differences in the selection directions?
You probably would. I just haven't looked at it.
Okay.
I do have, there's only one of me.
Yeah. Okay. I guess does the data even like?
Yeah, you could do this. You could do this. So, things like this. So what you get, metadata isn't great for a lot of the important things, but it does give you country and it does give you date.
Okay.
If you want to go into deeper things like, was this person vaccinated? Was this their first infection? Then you need to have, you can't do it with just say, they're trying to get more metadata, but it's reliant upon the submitters, which always means that, some people do a good job and some people do a bad job. Question?
Yeah, so my question is you're using a lot of DNDS-based methods, and my understanding is that they assume that synonymous mutations are neutral, but both non-synonymous and synonymous mutations could potentially affect translational efficiency. And I was wondering if you have done any work looking at, whether there are mutations that maybe don't have an effect on protein structure, but seem to still be spreading through the population that maybe are affecting something like translational efficiency.
There actually are, and it wasn't my work, but people have looked at, some of the synonymous. You have all kinds of weird things. You have in some of the proteins, for example, seem to be highly tolerant to things like frame shifts. So, there's like in the beta variant, there was a frame shift that cut off ORF8, like, two-thirds of the way through, and it was still fully functional. But there are some synonymous mutations that people characterize as important. We don't look at them directly. We do, our methods do correct for, synonymous rate variation, but we haven't looked at, the selective effect of synonymous substitutions. These methods don't really have the power to do it. You have to have additional information.
Dumb question, but a generic one. Do we expect the next variant to be less virulent or?
Well, do we expect the market to go up or down?
Turn off the video first.
Well, no, at this point, historically, when I was talking to Carl Zimmer, he asked me the same question, is like, what do you expect to find? And my answer to this is that we're trying to reason from a sample size that's small. We're trying to reason from a small number of recent historical outbreaks that we tracked. For most of things that we should know, if they became less or more virulent, we just don't know. For example, just to give you a sense of how little we know, measles, right? And measles is still What's the R naught for measles? The confidence interval is, I think, from 7 to 30. And the data that are typically used to infer this, one data set comes from World War I in soldiers, and the other comes from post-World War II Britain. That's what we base our inference on. So with this proviso, historically, what always happens, or tends to happen, the diseases that establish themselves into an endemic pattern, they try to find an evolutionary equilibrium, right? They can't infect too many hosts, kill too many hosts. So there's, and that generally trends towards sort of a lower virulence historically. But like with everything else, nothing's a given. Like it's probably, if you expect an endemic pattern, especially in a virus that jumps between hosts, can recombine, those reservoirs are going to be outliers. But, it's not going to be like, whatever, the Contagion, the movie, or, zombie apocalypse. So generally, I think we're just, and we got lucky with Omicron, right? Because it's basically done, exactly that. It's sort of changed enough function that it's now appears to be, a lot less deadly.
There's some questions on the Zoom chat.
Oh, right. Oh, apologies. Sorry. I need to bring my mouse to the right screen. All right, so I'm sorry, let me go through the questions. So if you're building a polygenic strain tree, how do you account for recombination events? So within SARS-CoV-2, there's not a whole lot of intra-strain recombination yet, so we can get away with it. But otherwise, we do this partition approach. So we take we use tools to screen for recombination, find recombination breakpoints, chop up the alignment, and for each putatively non-recombinant fragment, you do a separate tree. Is it possible that Omicron is a result of directed evolution experiment in mice? Everything is possible, but I have no idea what the relative likelihood of this is. And there is, I mean, you can make, in order to address this, you need to get more data from animals, and I think more data from chronic individuals. How does the predictor do in the mutations found in Omicron, given the rarity of those? Great question. When you have a sort of a shift, like an antigenic shift event, prior predictors break, because what used to matter is now in a different genomic background. So we have to wait for some Omicron data to accumulate for it to start working. Again, for the, oh, there we go. Then there's a nice reference for SARS-CoV-2 variants in patients with immunosuppression. Yes, so there are different types of immunosuppression, genetic disease, cancer therapy, or, just, pharmaceutical immune suppression or infection-based immune suppression. All right, thank you very much.